Hello everyone. Today our topic of discussion is on methionine metabolism. So in this video we are going to see all the exam oriented points on methionine and its metabolism. So this slide is the overall summary of methionine metabolism. So what is methionine? So methionine is a very essential amino acid and it is a glucogenic amino acid and it is also a very important sulfur containing amino acid. So when we see the structure of methionine, it has a very stable CSC, that is nothing but a thioether linkage, which makes the methionine very stable. So the first step in the metabolism of methionine is activation of methionine to SAM, that is S-adenosyl methionine. So it is a active methionine and it also acts as a principal methyl donor. So how this S-adenosyl methionine is formed from methionine? That is with the help of the enzyme called as methionine adenosyl transferase. Why it's so called adenosyl transferase is it transfers the adenosyl group from the ATP to the sulfur atom seen in the thioether linkage of methionine. So now in SAM, due to the presence of high energy bond, the methyl group in it has now become very liable and it can be easily transferred to other acceptors. So after donating the methyl group to the acceptor, the S-adenosyl methionine will result in formation of S-adenosyl homocysteine, and this happens with the help of the enzyme methyl transferase. So any acceptor that accepts the methyl group become a methylated acceptor or a product. So now the S-adenosyl homocysteine, by the action of the enzyme adenosyl homocysteinase, they result in formation of homocysteine. So what happens here is with the addition of a water molecule, the adenosine group is removed from S-adenosyl homocysteine. Finally, it forms homocysteine. So when we talk about homocysteine, it has two important fates. One is it can either synthesize cysteine or it can again resynthesize methionine. The one fate of homocysteine is formation of cysteine, and this happens by a two-step process. So in the first step, what happens is homocysteine, when it combines with serine, it results in formation of cystathionine, and this happens with the help of the enzyme cystathionine beta synthase, and B6 acts as a coenzyme for this step. And in the second step, cystathionine is acted upon by the enzyme cystathionase, finally forming cysteine. So the important clinical correlation of this cysteine formation is when there is deficiency or absence of enzyme cystathionine beta synthase. So these two steps are not going to take place, and finally what happens is this homocysteine get accumulated in the body, resulting in formation of homocystinuria. The next fate is resynthesis of methionine. So now what happens is the homocysteine, it can be converted to methionine by the addition of methyl group, and this methyl group is donated from the 1 carbon pool with the help of the coenzyme vitamin B12. This slide clearly shows how a methyl group from the 1 carbon pool is finally donated to homocysteine and how resynthesis of methionine takes place. So the methyl THFA from the 1 carbon pool is carried to the vitamin B12, and after donating the methyl group, this methyl THFA become THFA, and the B12 after receiving the methyl group, it becomes the methyl cobalamin, and from them the methyl group is finally donated to homocysteine and it helps in resynthesis of methionine. So in case if there is any deficiency of vitamin B12, all these steps of the reaction is not going to take place, and that's how folic acid is getting trapped, and this condition is called as methyl folate trap. So whenever there is deficiency of vitamin B12, we can also expect a simultaneous folic acid scarcity or deficiency. So next we are going to see about the few exam oriented points. So one is what are the two important fates of homocysteine. So one is resynthesis of methionine and then synthesis of cysteine. And next MCQ can be asked from the folate trap. So what is the vitamin associated with it, and how the folic acid is trapped? It can be trapped only in the form of a methyl derivative. And what are the important vitamins that are involved in the sulfur containing amino acid metabolism? So B12 and folic acid we have already seen in folate trap, and also B6. So B6, it is linked with the enzyme cystathionine beta synthase and also cystathionase, and B12 and folic acid is favoring this step, so that is catalyzed by the enzyme methionine synthase or homocysteine methyl transferase reaction. I hope you all understood the overview of methionine metabolism. So if you have any topic of interest to be discussed in the next video, you can put it in the comment section. So we'll see you all in the next video. Happy learning.